Thank you so much, Neil. And finally, I'd like to invite Charles Sawyers to the podium. Dr. Sawyers is the chair of Human Oncology and Pathogenesis Program at MSK, a dear friend, and I will never forget our first intro to AACR Genie, which he's going to talk about today, when he likened it to a Hamiltonian moment.
Thanks, Mia, and I'm really proud to be part of this panel. I'm really a cancer biologist and experimental therapeutics person and interloper into population science. Here's my disclosures. I can probably skip this, but maybe I'll just take a minute. We all know why we're here. Cancer is way more complicated than it used to be. It's hundreds of diseases, and some time ago I realized that we're going to have to organize ourselves in a slightly different way, and in order to make precision medicine drug development efficient. I think we learned that with the development of Herceptin, a long time ago. Hercept test. So Genie, which I'm going to tell you about, an important founding principle is that, and this is my personal belief, as maybe it's a lot of others too, is I think this information has to be pre-competitive. It shouldn't be held in silos. It's the sort of soil on which commercial development then depends, much like the NIH invests in basic science to foster drug development. And the last point is something I knew, but I've learned a lot more in sort of part of my oversight of Genie is the stewardship required. The history of me getting involved in this is I co-chaired a similar type conference, or it was actually a task force that met over a year that published this report. And I'll take a little bit of credit for the term precision. We actually had a whole 2-hour discussion at this meeting about whether to call it precision or personalized. I'm glad it stuck. And Kojo, I was interested in your earlier slide because this is the first slide in that report. And it's, of course, diagnosis is critical. All right, so the beginning of Genie. Yeah, you're right. Well, it was launched in 2015, but before that we had a series of pre-meetings. And I can't remember the first time that Hamilton was shown at the Public Theater in Manhattan, but I was lucky enough to get to see it. And it was like the founding of the colonies and all that, and I was like, this is what we're trying to do here, and here we are in Philadelphia at the headquarters of ACR, and there's Independence Hall. So that was the reference. The bottom line, we started, like anything like this, you can wait till all the sort of harmonization of ontologies happens, and then you can do something, or you can just start. And so we decided we would just start. It was 8 cancer centers that were willing to sort of pony up. And we dealt with, at that time, 8 different sequencing panels. And we decided that perfection is the enemy of the good enough, so we harmonized discordant panels. And that happened through a formal process at Sage Bionetworks. We decided to start small in terms of the clinical data elements. I'm not going to go through the history of it, but one thing I am proud of is we agreed amongst very passionate pathology groups from different institutions that a nomenclature called OncoTree would win the day, and that was a homegrown product. So here's what Genie is today. It's heavy on genomics, and it's getting much heavier on phenomics. We're about to hit 150,000 sequenced tumors. It might be there already. We're 18 centers now. We're announcing 4 new centers soon to expand diversity, and I'll explain why. I'm proud of the numbers at the bottom. That's how many people are registered to use the site and use it regularly, and there's a lot of publications that have come out over the 8 years. We have a splash page. This is through CBio Portal, developed by Nikki Schultz. And this was from several months ago, and it's figure 1 in a recent paper describing the first 100,000 patients' genomes. So for the clinical data collection, we've come a long way since that initial meeting, and we've invested heavily in collecting longitudinal data. And we are doing it in a combined structured data, algorithms that you're all familiar with, as well as curation manually of unstructured data using an approach that Deb Schrag had developed when she was at Dana-Farber called PRISM. I'm happy to answer any questions about it. That's now been rolled out at 4 institutions, and it collects, and we get these data elements here. And this biopharma collaborative, which is funded by 10 pharma companies in a pre-competitive way, has been critical at paying for that. It took a while to sort of get it going. Now we'll be at 16,000 patients by the end of the year, and it's going much faster than it was, obviously, in the beginning. That's across 4 sites, and we're doing now 8 sites. So I wanted to show you an example. So now that we have that data, we can do some really critical gut check experiments. And one is, how good is Deb's vision of determining progression-free survival using PRISM? So we were lucky enough to have RESIST measurements, and actually have bought RESIST measurements, and it looks pretty good. And there's 2 different, we can talk about this more, but there's 2 ways that PRISM scores PFS. One is by imaging, and the other is by a note from the medical oncologist that triggers progression. And they're pretty good at matching RESIST. This is just one example in lung cancer patients treated with checkpoint inhibitor. There's about 6 or 7 other examples I could point you to that Ken Keel at Dana-Farber has generated, as well as Greg Riley and Kathy Panagis at MSK. So what can you do with Genie data? Well, the size of it allows you to go after what we envisioned in the first place, and that is, rare genotypes. And rare genotypes, these are not rare across large, populations. They're just rare within certain, histologic definitions of disease. KRAS G12C is not exactly rare, but its approval was on single-arm studies by Amgen for sotorasib. And we're very proud that Genie data was used together with Flatiron Foundation data as part of the synthetic control, to lead to that approval. We've generated many other such examples, no other example yet of being used in a registration package. And we make this data, we publish it, we make it freely available. And I'm just proud of that one example because it shows that the data is robust enough to at least pass muster with the FDA. I want to end with something that is a new place we're going with Genie, and that is to explore race and ethnicity differences across cancer genotypes. There have been a number of publications using Genie that have made some really interesting observations about different frequencies of, for example, BRCA2 mutations in African Americans versus Caucasian men with prostate cancer. There's other examples in the literature, but I want to share 2 things that are from colleagues that are part of a program project grant that we've submitted to the NCI. One with implications for tumor mutation burden, and the other for cancers in which we don't have a driver identified by the sequencing panels that are routinely used. So I don't want to get into the weeds of this. This is work from Ryan Hernandez, Sasha Gusev, and Jian Karat Zhang at these 3 institutions working collaboratively, which is another thing I'm proud of, is Genie's brought together people across institutions. So the far left is MSK data in which we have tumor normal data. So we actually know when a variant is a true somatic variant because we have ground truth. But all the other panels in here do not have matching normal. And they're making variant calls based on reference databases. And the point is that if your variant allele frequency is up around 0.5, that means it's likely germline and not somatic. And you can see that with MSK, there's ground truth at around 0.25. The others, the Caucasian populations are lower down, but the others have higher VAFs. So there's clearly errors or miscalls. So what are the implications? Well, there is a test that you're all familiar with called tumor mutation burden, and it's calculated. The foundation version of this, and I'm sure you'll back me up on this, doesn't have matching normal, so it's calculated against a reference standard. So they ran a pilot analysis using truth from the MSK data of TMB low versus TMB high in outcome with checkpoint blockade. And basically the point of the blue line is that these were falsely called as TMB high, but they behaved as TMB low in terms of their treatment. So we're going to try to use examples like that to further learn and inform. The last example is I find shocking almost, and that is that in a cohort of patients who are called clear cell carcinoma, renal cancer, the frequency of VHL or even PBRM1, which is clearly a mutational alteration, not weird deletions or silencing, is much lower in blacks and African Americans than whites. And there's that plot on the right is an admixture analysis making it clear that this isn't environmental, this is due to a genetic background or genetic ancestry. So why is this? Well, I think the sequencing panels that we've optimized have been based on cancer registry data that are largely European. And so we're whole exoming all these patients to see what happens. So I'm going to close with what I think is sort of a challenge that, I think Kojo, you and Hedy both raised it in the opening, and that is how do we can get really good at doing this, and like Neil, I think we're ready. I mean, but we have to make sure that the data is generalizable. So what is Genie doing? I mentioned we're adding 4 new sites, and whether they are really going to address that in a major way, this gap, we'll see. We've actively invited collaborations with groups that have more diverse populations with genomic data. Number 2, Dr. Carrot Zhang has developed ways of determining genetic ancestry from the existing panels. So we're doing that across the entire Genie registry. So that's going to be available for 120,000 cases. We already know the differences in cohorts that we've looked at from self-reported race, and we can see admixture analysis, the power of that, as I just showed you. But I want to finish with this, what I think is the barrier for, even though we want to include individuals of non-European ancestry, we have to make sure they're properly consented, or we have to come up with new ways to, take care and sort of administer this data fairly. And that's why I was interested in the consent question, a tool from you, and I was very interested, Brad, in your digital, patient direct consent methods. I think we're going to have to figure out ways to incorporate that going forward, to bring in these populations, which quite frankly, there's a lack of trust that I've even run into already in setting up some of these collaborations. So thank you.